Hello and good afternoon friends, welcome to the CEC-UGC live lecture. Dear friends, with our ongoing series on medical diagnostics, we have conducted numerous sessions. Dear friends, in our previous session we talked about vaccines. We tried to understand what vaccines are. In today's session we would be discussing on different types of vaccines, and for this very discussion we have once again with us in our studios Dr. Hardeep Kaur. Dr. Hardeep Kaur is Assistant Professor in Department of Zoology, Ramjas College, University of Delhi. So let's welcome our guest Dr. Hardeep Kaur, and let's try to understand more and more about vaccines. Hello ma'am, welcome to the UGC lecture.
Thanks a lot. Hello viewers, once again welcome to this second session on vaccines. Now let us recall what we did in our first session. Now, if you remember in our first lecture, that is vaccine part 1, we learned what exactly are vaccines. Now if we just want to recollect the definition of vaccine, it is a biological preparation that improves immunity to a particular disease. It typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed form of microbe or its toxins or one of its surface proteins. Now viewers, if you remember that we had defined what exactly are vaccines in our last lecture, we went into the details of it. We realized that technically it's a kind of an infectious organism. We have modified it, we have attenuated it, weakened it, or maybe killed it, or we had tried to use any of its product as a vaccine mechanism. Once we had used it, the body starts producing an immunity against it, and once this immunity is produced, now our body is ready to actually combat the real infectious agent. So with this, let's go further. In our last lecture, we also went into the history of vaccination. Of course, we discussed how the Chinese and Turks initially started with the process of variolation. Following that, we gave an in-depth study of how Edward Jenner came up with vaccination. Now, if you remember, during let's say 18th century, the end of 18th century, Edward Jenner, who was an English doctor, he actually learned from his milkmaid that since they were inflicted by cowpox, they were protected from smallpox. This theory he actually worked upon. He was trying to be a physician himself. So he realized this theory, and then what he did was to authenticate this theory, he actually inoculated one of his subject, that is Edward Phipps. This we had discussed in our previous lecture. He actually inoculated cowpox pustule from material from the cowpox pustule into Edward Phipps, and Edward Phipps was eventually protected from smallpox. So this was the first authenticated case of vaccination. No wonder we call Edward Jenner as father of immunology also, and he is remembered as the pioneer of smallpox vaccination. Now of course we cannot undermine the work done by Louis Pasteur. Louis Pasteur was the first one who came up with he actually found out the causative agent for cholera in fowl. Later on he actually found out the vaccination against rabies and anthrax, and he has done pioneering work in immunology and vaccination. His discoveries of vaccination, microbial fermentation and pasteurization are well known. Now we cannot again undermine the work done by Dr. Maurice Hilleman. Nearly 40 different vaccines were the outcome of his work, scientific work. So no wonder he is called father of modern vaccinology, as he developed almost, as I just said, 40 vaccines. Now these were the pioneer people. In our last lecture, we also did an overview of the immune system. We realized that the pathogen could be a foreign particle, a virus, bacteria, parasite, fungi, anything. Anything which is not belonging to your body, we call it as an antigen. Now whenever an antigen enters our body, there is a first line of defense which is by the phagocytosis. The phagocytes could be macrophages, neutrophils, anything which actually who do not discriminate between what the pathogen is. For them, it is like they just would be eating or would be phagocytosing whatever pathogen is entering. That is a first line of defense. Following that, a proper adaptive acquired immunity gets initiated maybe once the primary line of defense fails, the secondary line of defense, which is actually a very specific immunity, comes up, wherein we have our soldiers like T helper cells, the T cytotoxic cells, the B lymphocytes. All of them, for example, B lymphocytes produces antibodies which are exactly specific to a particular antigen. Now, this comprises of our immune system. Once a pathogen enters our body, our immune system gets activated, these cells get activated and they produce a very specific immune response against this pathogen, so much so the pathogen is being killed. And not only that, these pathogen, there is a memory associated with this kind of a specific immunity. So, the body also memorizes this. With having understood this concept, we went on to define vaccination. Actually, what we are doing is vaccination is just following exactly what is happening naturally in the body. Now, with this, let us see this flow diagram here. As I told you in my last class, in my last lecture, immunity is of two types: innate immunity, adaptive immunity. Adaptive immunity further can be of natural adaptive or artificial adaptive, and further natural can be natural active, natural passive, artificial active or artificial passive. If you remember, when we did natural active immunity, adaptive immunity, we realized that this is when a pathogen, any pathogen comes inside our body, the body gets disease, the body undergoes a disease, and now the body has, the immune system has got activated and the immune system has produced certain memory cells. So, the same pathogen, if it comes back again second time, the body is already prepared. Now, the same principle is followed in the artificial active immunity. What happens is, here what we do, we are using a attenuated or a weakened pathogen and exactly imitating what happens. So, the weakened pathogen is given in a lower dose inside the body. The body or the immune system produces a response. Now, this response is there, it leads to a memory of this pathogen. So, the actual virulent pathogen or infectious agent comes next time, the body is already prepared. That is what is followed in vaccination. Now, how vaccines prevent disease? Here, as you can see in the example, vaccine contains the same antigen or the part of an antigen that causes disease. So, vaccines are nothing but an attenuated pathogen or a weakened pathogen or a killed pathogen. Live, attenuated, killed or a part of the pathogen. Now, for example, here as you can see, the measles vaccine contains measles virus. But the antigen in vaccines are either killed or weakened to the point that do not actually cause a disease. Now, as you can see, the blue one is the measles virus. The part of this measles virus or maybe the entire measles virus is being attenuated or weakened and is used as an antigen in the vaccine, and as you can see in the body, in the blood vessel, you can see the blue colored Y-shaped antibody which is actually specific to this particular antigen being the measles virus. This is the principle how vaccines actually prevent diseases. So, once these antibodies are formed, when the real virulent or activated measles virus will come, eventually inflict this organism, the organism is already prepared because he has already got a pre-formed antibody against the measles virus. So, this is what happens in vaccination. Now, does the vaccines work? Of course, the vaccines work because you can see it from this table. As you can see, before vaccination, the number of cases in 1975 of smallpox were almost 23,546 per year. But after vaccination, there was 0 case in 1976, 100% eradication of smallpox. Similarly, if you see for polio in 1980, there was around 18,975 cases which got completely eradicated in 2012. No wild polio virus has been detected since 2011. Similarly, for other cases, for example, in measles there is an 84% decrease, for pertussis there is 86% decrease, diphtheria there is 94% decrease post vaccination, for tetanus there is almost 95% decrease both neonatal and as well as in adults. So, we can very well say yes, vaccines do work, right. So, with this, let us come to the real topic of today's discussion, the major types of vaccine. This slide is actually, yes, in the last lecture also we started with this, the major types of vaccines. we said that major types of vaccine include the whole organism vaccines, where the entire organism is being used as a vaccine, you know. The entire pathogen may be attenuated, that means weakened due to treatment, or it is actually killed. So, if it is killed, there is no way it is going to cause infection in the person in which you are putting it. But if it is not killed, it is live, then there is a chance that it might cause an infection in the organism in who is receiving the vaccination. So, in that case, we weaken this pathogen to a level where it does not lead to a disease, or it might lead to a very mild manifestation. So, this is called a whole organism vaccines. Now, there are purified macromolecules which are also used as vaccines. Now, if you remember, we said that toxoids which are secreted by certain pathogens can also be used as vaccines. The capsular polysaccharide, that means the polysaccharide coat, can also be used as a vaccine. The recombinant microbial antigens or the surface antigens can also be used as a vaccine. There is a new class of vaccines called recombinant vector based vaccines, the DNA vaccines, the synthetic peptide vaccines, and the multivalent subunit vaccines. We will be discussing all these major types of vaccines in our today's lecture. Now, coming to the first one, that is the whole organism vaccines. As you can see on the left hand side, it is a whole inactivated, the whole organism is being used. Here in this case, it could be a bacterial cell or it could be a viral particle, but it is a complete bacterial cell or a complete virus particle. On the left side, you see the blue one is in totally inactivation of the pathogen has been done. It could be done by heating the pathogen or by certain chemical means, or it could also be done by other means, so that it is no longer capable of replication in the host. So, there is no danger that once given into the person or the host will not show any manifestation of the disease, because here the pathogen is totally wholly inactivated. Now, on the right hand side, you see it is a live attenuated pathogen. The pathogen is live, but it has been attenuated. Attenuation means it is like weakening of the pathogen, which can be achieved by growing a pathogenic bacteria for maybe even a virus for a prolonged period under certain abnormal culture conditions. So, you give certain different culture, or you start depleting culture, so that finally what you get is a very weakened pathogen. So, as I just said, if the pathogen is weakened, chances are that it is not capable of causing the disease in the host now. But maybe even if there is a very slight chance, then it will be a very less manifestation, maybe a very little fever or very less manifestation of the disease. But a disease may not be there unless there is something really wrong with our vaccine. So, technically live attenuated vaccines are generally safe. But of course, off and on you do get cases where the live vaccine is actually being the cause of the disease. So, unfortunately those cases, otherwise the live attenuated vaccines are equally safe. Now, let us go into few of the examples of whole organism vaccines. Let us understand the live attenuated avirulent vaccines. So, the first one was against the tuberculosis disease. Here we are using a live but attenuated BCG vaccine, Bacillus calmette-guerin vaccine. For typhoid, again we are using a live attenuated vaccine. Both of these were discussed in our previous lecture. So, for the other ones, we also discussed the polio vaccine, the Sabin one, the live attenuated Sabin vaccine, which was again discussed in part 1. For the other ones, for example, for other viral diseases like measles, mumps, and polio, the Salk one, the rotavirus, the varicella zoster, yellow fever. Again, the type of vaccine is a live attenuated. That means the live vaccine is being used, but it has been extremely weakened. It's been attenuated. So, let's come one by one to these vaccines. The first one, let us discuss the measles. Now, it's a live attenuated measles vaccine, and you know that measles is an acute illness which is caused by a virus, which is paramyxovirus family. It belongs to paramyxovirus family. It is transmitted via droplet from the nose, mouth or throat of the infected persons. As you can see, the child is actually suffering so many blisters, there are so many manifestations, there were so many blisters on the body of the child. Although it is not as big as the smallpox, there are smaller ones. The virus infects the respiratory system, making it highly contagious. So, technically it is kind of a droplet, spreads through a droplet infection. Globally, an estimated 450 people, mostly children, die every day from measles, despite the fact that an effective and safe vaccine is actually available. Still, every day globally 450 people, mostly children, are dying due to measles. Now, as you can see here, what is happening, it is a preparation of the measles vaccine. How it is being prepared, since it is a highly contagious virus, you can see all people who are preparing it, they are wearing masks and all in the laboratory. Now, the first measles vaccine which was tested was by Sam Katz. He is an MD. He is an infectious disease specialist who was working with Thomas Peebles and other researchers in the Boston lab. He tested the first version of the lab's vaccine on mentally retarded and disabled children at a school outside of Boston. Each of the 11 vaccinated children developed measles antibodies, but 9 also developed a mild rash. The vaccine did not cause full blown measles, but it did cause little symptoms. The researchers realized that the virus used for the vaccines had to be weakened even more. This is the first case in 1958 where Katz actually tried the first vaccine against measles, and since this vaccine was still not ready, it was tried in 11 kids, and out of whom 9 actually developed small rashes on their body. So, that means they realized that this vaccine needs further improvement and it needs further attenuation or further weakening. After that, in 1962, attenuated measles vaccine developed. Maurice Hillman, the person who is the father of modern vaccinology, and his colleagues developed an attenuated measles vaccine by passaging John Enders measles virus strain over 80 times through different cell types. The resulting vaccine, which is called Ribuvax, was given with a dose of gamma globulin antibodies to reduce reaction, that is mainly fever and rash. Of course, this was a very successful vaccine. Now, Hillman is also responsible for creating the vaccine against mumps. Now, it was known that his daughter Geraldine actually developed this mumps, she was afflicted with mumps disease. Now, mumps is caused by, again it is a viral disease, but it is a very contagious disease. So, when his daughter got mumps, what he actually did, he actually got swabs from the mouth of his daughter, and he actually cultured it, and later he used it to vaccinate his younger daughter also. So, as you can see, mumps is a contagious disease which is caused by a virus that leads to painful swelling of the parotid gland. Parotid are the type of a salivary gland. So, mumps mostly commonly occurs in children between the age of 2 and 12 who have not been vaccinated. Symptoms can include fever, headache, painful swelling, swollen glands in face, neck and jaw. As you see, it is totally afflicting the salivary gland. So, naturally there is a lot of pain in the mouth, on the sides and all. So, Holman, what he did, he isolated mumps virus from his daughter. He believed that since she had mumps, so she must be having the virus in her mouth. So, actually he took out the swabs, as I just told you. He swabbed Geraldine's throat and placed the swab in the broth, and hence he cultured it and then kept it in his lab, and later used it as a vaccine to vaccinate his other daughter. Here, as you can see, the other daughter, the Geraldine is the one, the elder daughter, and the younger one is the one in whom this vaccination is being given. The younger one is Kirsten. So, Hillman arranged for his daughter Kirsten to receive his experimental mumps vaccine, which was developed from the virus isolated from her half sister Geraldine. So, this is how he actually worked on mumps vaccine, first on his own kids. Now, other whole organism vaccines that are inactivated, that is killed vaccines. Here Till now we were doing the attenuated or the weakened vaccines, where there was still a danger that it can cause slight manifestation of the infection. But here what we are talking about, those vaccines where the pathogen is totally killed. Now few of those whole organisms killed vaccines are against anthrax, here where we are using an inactivated vaccine. These are all bacterial pathogens, anthrax, cholera, pertussis, plague, they are all caused by bacteria, and the type of vaccine which is used to take to combat this disease are the inactivated or the killed vaccines. For against the virus particles, especially like hepatitis A, influenza, polio, the Salk one. Rabies, rubella, again we are using an inactivated or completely killed vaccine. So let us come one by one to these ones. Who will forget the anthrax, the use of anthrax in the biological warfare? I mean, nobody can forget the 9/11. These are few of the envelopes which were sent to congressmen and news agencies that actually contained anthrax spores. This is those letters which were sent post 9/11. Now, the threat of infection from potential agents of bioterrorism are immense right now. Anthrax topping the list. So, anthrax played a anthrax actually has a real role in a biological warfare mechanism. Now, infection due to anthrax occur by 3 different routes. First of all, inhalation causes severe flu-like illness with high mortality rate unless it is diagnosed and treated immediately with antibiotics like penicillin, doxycycline or ciprofloxacin, otherwise the mortality rate is very high. The cutaneous exposure results in skin lesions with characteristic plaque deep eschar. Cutaneous anthrax has a 20% mortality if untreated, but usually responds to antibiotics. The GI exposure, that is the gastrointestinal exposure, results in ulcers in the ileum or cecum, bloody diarrhea and sepsis, and is almost always fatal because generally it goes undetected and diagnosis is not made at the right time. So, most of the times people die because of anthrax. Now, how anthrax vaccine actually has been made? The anthrax vaccine is made by taking a strain of bacteria Bacillus anthracis and growing it in the laboratory. Now, the bacteria releases certain harmful proteins which are called the toxins, which actually come into the surrounding broth. So, these toxins are actually responsible for the disease. So, what the scientists did, they took the toxins, inactivated them with formaldehyde. Now, inactivated toxins are called toxoids. Now, these toxoids are used as a vaccines. These toxoids are actually, so technically we are not using any cell of the bacillus, we are directly using the toxin which is produced by the bacillus. So, it is a kind of a acellular vaccine. Now, let us go to the next disease, which is plague. Now, as you can see, the plague there was an outbreak of plague in India in 1994. It was almost of an epidemic proportion. So, this is a Newsweek that week where it says plague, will it spread? It was really rampant in 1994. Now, the transmission of plague, if you know, the plague is caused by Yersinia pestis, which is a gram-negative bacillus and belongs to the family Enterobacteriaceae. Now, as you can see, the infected rodent is one of the intermediate hosts, and then the rat fleas who feed on the blood of the rodent, which when actually infect human host or bite human, spreads the disease into human as well. So, technically rat fleas are responsible for transmitting disease from the rodents to human in case of plague. Now, there are 2 types of plague we all know, one is called the bubonic plague and the other is called the pneumonic plague. Now, if you remember, the bubonic plague is actually spread because of the rat fleas. The rat fleas feed on the blood of human. The pathogen, that is the pathogen of plague actually enters into the lymphatics and the systemic circulation of the human. It finally causes disease, leads to buboes, which is black hemorrhagic lymph nodes, and pneumonia and internal organ hemorrhage within human. And finally, it exits by droplet infection, by sneezing or by any of the by sneezing, coughing, etc. And as I just said, it is a highly infectious disease. On the other hand, the pneumonic plague has the same entry and exit through, of course, it's a droplet infection. Again it is highly contagious, but here mostly the pathogen is localized in the lungs. Mortality rate specifically in case of pneumonic plague is almost 100%. So, you can realize that how plague must have devastated human life in 1994 in India. So, what the plague vaccine, what is the history of the plague vaccine? The prophylactic vaccination against bubonic plague was started in India. It was Dr. Waldemar Mordecai Haffkine. When the plague pandemic reached Bombay, Dr. Haffkine became bacteriologist to the government of British India, that is between 1896 to 1915. He soon produced an effective anti-plague vaccine. Now, it killed bacteria was used in plague vaccine since 1896. After that, in the pandemic of Asiatic cholera in 1817 to 23, it was seen this was the first great cholera pandemic in the 19th century. It was unprecedented in its fury, affecting almost every country in Asia. While early cases of cholera were reported from Purnia, that is in Bihar right now, in early 1816, the pandemic is believed to have originated in the town of Jessore near Calcutta in August 1817. A civil surgeon reporting on the high incidence of a severe gastrointestinal disease among his patients drew attention to the source of contagion, that was the contaminated rice that was consumed by them. Amidst various attacks of vomiting and diarrhea, of thousands of people collapsed and died, including hundreds of British soldiers transmitting the disease throughout Bengal. Later, cholera spread into Sri Lanka also. Now, how cholera? Koch was the first one who isolated the cholera bacillus. He, in a letter from India describing his research, announced his discovery of the agent that causes cholera. For a time, it was called the Koch bacillus or Koch bacillus, because that is the shape of the bacillus. Following that, Ferran's vaccine came up. He was a Spanish physician who developed a cholera vaccine. His vaccine was first to immunize humans against a bacterial disease. Ferran had worked on veterinary vaccines in Spain following Pasteur's publication on attenuation of microbe. Hence, he created a cholera vaccine by attenuating the cholera bacillus. He grew the cholera bacteria on a nutrient medium and room temperature. The material was then administered to subjects via one of the 3 injections in the arm. Now, with this, we will come to the end of the first session. We will continue with vaccines in our second session. Thank you so much. Welcome back, viewers. We were discussing about the various types of vaccines. We had already discussed the live attenuated whole organism vaccines. We were actually discussing the whole organisms killed or inactivated vaccines. In our previous session, we discussed about anthrax, we discussed about cholera, we discussed about plague. Let us come back to the viral particles that have been inactivated and used as a whole organism vaccine. So, we will be discussing about polio, which was a dreaded disease during some times back. Of course, we will talk about rabies, influenza and hepatitis A. So, coming first to polio, so it was actually Jonas Salk who came out with the Salk vaccine. Of course, there was a Sabin vaccine also, if you remember, we discussed in our part 1 where we actually discussed what is a Sabin polio vaccine. Now, Salk polio vaccine was a totally inactivated vaccine. Salk and his team, with the support of National Foundation for Infantile Paralysis, began its first test on humans of the killed virus polio vaccine. These subjects were resident children in institutions for the disabled and retarded. Salk tested vaccine for all 3 strains of polio, some in combination and some on their own. Now, their findings showed that the vaccine recipient produced antibodies to the virus in the vaccine which was given. You must understand there are 3 different types of viruses in this case. So, and they found that when given in combination, all 3 different types of antibodies were produced within the host or the people who received the vaccine, the recipients. The Salk results provided some evidence on which later trials were based. Now, with this further, as you can see in this slide, in 1953 here Salk is giving vaccine to his own family. This Mrs. Salk, and of course, this is one of the 3 sons of Salk, and they are getting this experimental polio virus vaccine. Now, in 1955, in a press conference at the University of Michigan, Thomas Francis Jr. MD, a scientist with extensive experience with influenza vaccine, and his colleagues announced the results of the Salk polio virus vaccine trial. The vaccine, they said, was 80 to 90% effective against paralytic polio. The US government actually licensed Salk's vaccine later the same very day. The press conference and licensure paved the way for widespread distribution and use of this vaccine. Unfortunately, it has to be suspended that year itself because Leonard Schill, an MD and the US Surgeon General, actually suspended the polio vaccination program in order to investigate the safety of all 6 manufacturers' vaccine. So, the full review which was done by Schill, it concluded that 11 people that died from the vaccine and 100 of people were paralyzed. Though the cause of the disaster was never proven, it is likely that certain production methods, which actually it turns out did not follow the Salk's instruction. That was the cause of disease in few of the subjects. This resulted in failure to completely kill a certain type 1, which is called Mahoney type of the virus, which resulted in polio in those subjects or in those individuals that still received the Salk vaccine, and they got polio because of the vaccine. So, in 1955, for instance, insufficiently killed virus in the vaccine from Cutter Laboratories in Berkeley, California infected some 200 children. Many were paralyzed and several actually died. So, technically the virus which was used in vaccine in that case never got completely killed. So, the very important thing as described was by Salk was to completely kill or deactivate the virus, which was not followed nicely by Cutter's laboratory, which actually led to infection in various children and eventual death of those people. Now, later for rubella and rubella vaccine, it came out in 1740, it is called German measles. Now, there is a reason why it is called German measles. German physician Friedrich Hoffmann was the first to give a clinical description of the disease that later came to be known as rubella. Until that point, however, so many German physicians were involved in identifying and distinguishing the disease from other known maladies after Hoffmann's description. German physician de Bergen and Orlow confirmed his work, and that it would be referred to as Rottelin from German word Rottelin. Thus, rubella eventually came to be known as German measles. Now, rubella is again caused by a virus. The first time rubella vaccine that got licensed was in 1969. Meruvax, that is Hillman's HPV 77 rubella vaccine, was licensed. Later it was replaced by Stanley Plotin's RS27/3 strain of rubella virus. What exactly is rubella? Rubella is a very mild disease. Its symptoms are low fever, rash, swollen glands. However, in a pregnant woman, if she gets inflicted with rubella, it almost always causes devastating effects in the fetus. The babies exposed to rubella in utero may suffer from deafness, blindness, mental retardation and other deformities. In the United States, more than 20,000 babies were born with congenital rubella syndrome. Tens of thousands of women suffered miscarriages or elected to have abortions due to rubella infections. The pandemic focused medical researchers' attention on the need for a very effective vaccine which later came. Now after that a new vaccine came up which is called MMR, still kids are given MMR. The MMR stands for mumps, measles, rubella vaccine. So it is a combination vaccine. Now MMR combination vaccine came up in 1971. Of course, it was due to the efforts of Hillman again. The US government licensed Merck's measles, mumps and rubella, that is MMR combination vaccine in 1971. So in an article published in the Journal of American Medical Association, researchers reported that the vaccine induced immunity to measles in 96% of vaccinated children, to mumps about 95% and to rubella in 94%. Additional tests which were done in 1968 also showed that the adverse reaction from MMR vaccine was no greater than giving it these vaccines singly. So whatever the side effects of vaccines for mumps or measles or rubella, if given singly, it is not increasing the side effect even when it is given in combination. So it was pretty safe vaccine which is still followed to this date. Now let us come to another type of vaccine where we are using the macromolecule of the pathogen, not the complete pathogen, but a part of the pathogen as a vaccine. So we are using a purified macromolecule as vaccine. So you must understand, viewers, that if the pathogen is not complete, it is just a part of the pathogen, maybe an encapsulation, the capsule protein or maybe some toxin. So technically we are not using the complete pathogen, and if we are not using a complete pathogen, then there is no point, no chances of getting a disease in the individual. So this was a much better form of vaccine. So let us come to the purified macromolecules as vaccines. In certain cases the toxoids are the toxins which are secreted by the cell of the pathogen, are used as a vaccine. For example, in case of diphtheria, it is the inactivated exotoxin, or in tetanus, it is again the inactivated exotoxin which is used as a vaccine. Of course, the capsule of the capsule polysaccharide can also be used as a vaccine. For example, in case of Haemophilus influenzae type B, polysaccharide and a protein carrier is used. In Neisseria meningitis, polysaccharide is used. In Streptococcus pneumoniae, 23 distinct capsular polysaccharides are used as vaccines. Similarly, the surface antigen in case of hepatitis B is used. It is a recombinant surface antigen which is used. We will discuss them one by one. Now, the toxoids are manufactured from bacterial toxins. Some bacterial pathogens, for example like diphtheria and tetanus, they produce certain exotoxins. Now, we can take out exotoxin. These exotoxin are actually responsible for the entire disease symptoms. Now, these endotoxin produce many of the disease symptoms that result from infection. Now, tetanus vaccine, for example, can be made by purifying the bacterial exotoxin and then inactivating the toxin with formaldehyde. So, once we inactivate the toxin with formaldehyde, we call it as toxoid. Now, this toxoid is safe to be used as a vaccine in an individual and is not going to cause any disease symptoms in an individual. So, with this we develop the effective vaccine for diphtheria as well as tetanus. Now, for example, the tetanus vaccine, as you can see in the diagram, here is the bacteria. In case of this Clostridium, the Clostridium actually secrete these exotoxins. These exotoxins are proteins which are produced inside the pathogenic bacteria, mostly gram-positive bacteria. As part of their growth and metabolism, these exotoxins are secreted into the surrounding medium, and finally they are harbored from the surrounding medium, then they are inactivated using formaldehyde, and hence the tetanus vaccine is made. Now, similarly for Haemophilus influenzae, as I just told you, for Haemophilus influenzae, the bacterial polysaccharide capsule is used as a vaccine. Hib, as it is commonly known, is the major cause of bacterial meningitis in children less than 5 years of age. So, generally the capsule which is encompassing the bacteria is taken out. It is not the complete bacteria we are using, it is only the polysaccharide capsule which we are using it as a vaccine. Now, here as you can see how we are doing it, as on this right hand side you will see there is a bacterium the enlarged view of the bacterium has been shown. The surface polysaccharide coating is taken out of the membrane of the bacteria. It is then linked to a carrier molecule, and that is how the vaccine is produced. So, vaccine for Haemophilus influenzae type B, that is HIP, consists of type B capsular polysaccharide, which is then covalently linked to a protein carrier, that is the tetanus toxoid. The polysaccharide protein conjugate is much more immunogenic than polysaccharide alone. It activates the T helper cell, enables class switching from IgM to IgG, and also induces memory in certain B cells. So, all in all, once you are taken out the polysaccharide, linked it to a carrier molecule, it is a very good vaccine. You put it into the individual, this vaccine activates the immune system, causes all important things, produces a memory, and hence the person is protected from this particular influenza bacterium. Now, in case of Streptococcus pneumoniae, again the bacterial polysaccharide capsules are again used as vaccines. It is the same process wherein the bacterial capsules are harbored from the bacterium, and these capsules may be directly used as a vaccine in case of human. Now, bacterial polysaccharide capsules are used as vaccines. For example, the Pneumovax, pneumococcal vaccine polyvalent, is a sterile liquid vaccine for intramuscular or subcutaneous injection. This vaccine protects against the Streptococcus pneumoniae bacteria that causes meningitis as well as pneumonia. It actually consists of a mixture of highly purified capsular polysaccharides from 23 most prevalent or invasive pneumococcal types. So, it is all 23 types of pneumococci. Their protein or the polysaccharide molecule is taken from their capsule and is given together in one vaccine. So, you are actually getting a protection from 23 types of pneumococci. Now, proteins from pathogens are produced by recombinant techniques also. Now, for example, the gene encoding any immunogenic protein can be cloned and can be expressed in bacteria, yeast or mammalian cells using rDNA technology. A number of genes encoding surface antigens from viral, bacterial and protozoan pathogens have been successfully cloned into bacterial, yeast, insect or mammalian expression system, and the expressed antigen is then used for vaccine development. Now, the first such recombinant antigen vaccine which was approved for human use is actually the hepatitis B vaccine. Now, how actually it happens in case of hepatitis B? Now, let us understand what hepatitis B is. Hepatitis B is a viral infection that attacks the liver specifically and causes both acute and chronic disease. The virus is transmitted through contact with the blood or other body fluids of an infected person. More than 7,80,000 people die every year due to the consequences of hepatitis B. Now, the hepatitis B vaccine is almost 95% effective in preventing the infection and its chronic consequences. Now, how exactly the hepatitis, this HB, Recombivax HB vaccine is made? Let us understand. It is a non-infectious vaccine which consists of a surface antigen, HBs antigen of hepatitis B virus produced in yeast cell. Now, if you see on the left hand corner, you have the hepatitis B virus which has a RNA material, genetic material. Now, the HB antigen produces gene. The genetic material, part of the hepatitis B virus genetic material is taken. Now, on the other hand, you will see on the lowermost end of on the left hand side corner, you will see there is a bacterium, bacterial DNA is, genome DNA is there, but there is also plasmid DNA. As we know that it is a self-replicating independent genetic material in bacteria, we harbor this plasmid DNA and cut it with the restriction enzymes. Generally, this process involves using the same restriction enzymes as it is used in case of hepatitis B, cutting the genetic material from the hepatitis B virus. Now, what we do is we ligate it. So, we ligate our genetic material coming from the hepatitis B virus into our carrier, which is a plasmid. Now, once this hybrid product is formed, once we have ligated our genetic, our desirable genetic material into a carrier here in this case is a bacterial plasmid vector, and then we introduce this recombinant DNA or recombinant plasmid molecule into a yeast cell. Now, recombinant yeast cell will be expressing that gene, that insert or that the viral genetic material, which we are calling it as an insert here, it will be expressing it as it is doing it for the other genetic material. So, once that gene gets expressed, it will produce a protein, and the protein will cause, this will be used, the recombinant yeast cell will multiply and will produce as HP antigen in the fermentation tank. We will extract out this protein, this expressed protein, and we will use it as a vaccine for our purpose. Now, coming next to multivalent subunit vaccines. Now, this is another very latest type of vaccines wherein incorporation of multiple antigens from the same microbe is done into the vaccine through the construction of solid matrix antibody antigen complexes by attaching monoclonal antibodies to particulate solid matrices and then saturating the antibody with the desired antigen. As you can see, we have a solid matrix on which we have various monoclonal antibodies. These monoclonal antibodies are specific to different antigens. So, we can take multiple subunits from a pathogen, put them as an antigen. See, since the antibody is specific to each antigen will bind to its specific antibody. So, we have actually created a matrix, a solid matrix having an antibody, specific antibody on its surface, and the antigen surrounding or getting combined to its specific antibody. Now, this itself can be used as a vaccine to actually vaccinate certain host organism, and the host will produce an antibody, a specific antibody to these antigens. Now, the subunit vaccines, first of all in 1996 it was produced the prospect for subunit vaccine against the tick-borne diseases. Following that, in 2011 the subunit vaccine was produced against malaria. Then of course, in 2012 a peptide based subunit vaccine was produced against hookworm infection. These are few of the research papers wherein it is being used, this concept of subunit vaccines was used to take care of the tick-borne diseases or the hookworm infection or even malaria. Now, again a very related way of producing vaccine is a DNA vaccines. As you can see in the figure, what happens, the encoded protein of the DNA vaccine is expressed in the host in its most natural form. As you can see, there is a pathogen which has a genetic material, the gene is taken from the antigen, it is put into the plasmid, and finally directly this vaccine is used rather than the product of this. Directly this DNA vaccine is used using a gene gun or even by injecting. So, DNA vaccines induce both humoral as well as cell mediated immunity. They generally cause prolonged expression of the antigen, which leads to a better immunological memory in the host. These are some of the latest techniques of vaccine development. Now, again let us see in this. This is a pathway of induction of immunity by DNA vaccine. As you can see, we have a DNA plasmid vaccine wherein we have a gene of our interest, which is the gene coding for the vaccine antigen. There is a promoter which has been attached to it, it has been put into a plasmid, and then finally there are two routes, either indirect route wherein it is put into a muscle cell or a skin cell by using a gene gun, or by using a intramuscular injection or a cutaneous subcutaneous injection, and then it is then cross presented, or directly it is put through an antigen presenting cell, and these vaccine contain only the portion of the pathogen genetic material which then instruct the human cells to make proteins that lead to an immune response to that particular pathogen. Now, with this let us come to a very important new invention which is called the cancer vaccine. It is a new feature, it is the immediate future. Now, as you can see the cancer vaccine design, how actually it happens, we take out a person who's inflicted, who's been suffering from cancer, we excise out tumor from that person, or use certain tumor cell line can also be used for purpose of laboratory. But otherwise, a person who's actually affected by a tumor, we took out a piece of or a tumor from that person, we isolate the dendritic cells from the same person. Then we mix the killed tumor cells or the protein extracted from the tumor cells with the dendritic cells from the same patient. And then these dendritic cells having the killed tumor cells are returned back as an antigen and are vaccinated back into the same human. Now understand, if the dendritic cell, which is nothing but an antigen presenting cells, is presenting a tumor cell to the body will start producing, the immune response will be produced in this individual, and he will start producing antibodies against this particular tumor cells. Now because tumor has been is found as an antigen in this case. Since it is a dead, it is not spreading the tumor cells are already dead. Now once the antibodies are produced, so these antibodies will roam around in the body and will tend to kill all the tumor cells wherever they are. So this is a new cancer vaccine design which has come out. Now this is how it works. As you can see, the tumor RNA is inserted into a dendritic cell, or even the tumor antigen is inserted into a dendritic cell. The dendritic cell matures, produces tumor antigen, which is injected back into the same patient. Understand, it is the dendritic cell from the same patient. So the patient is not going to consider this dendritic cell as foreign, because it is his own dendritic cell. Now this dendritic cell, which is already having the tumor antigen, will present the dead tumor antigens on its surface to the immune system, the T helper cells or the T cytotoxic cells. These T cells will further lead to a immune response production. It will give, if it is a T helper cell, it will give help to the B lymphocyte to produce a specific antibody against this tumor antigen, and wherever the tumor cell is there, these antibodies once produced will finally go and attack those tumor cells in that same person's body. Now of late, there is a lot of attention being given to Ebola. Ebola virus is actually affecting so much, specifically in the African countries. You will say especially Ebola symptoms occur maybe from 2 to 21 days after exposure, but mostly it has been seen between 8 to 10 days. Now the first stage occurs between 7 to 9 days. There is a lot of joint pain, sore throat, fever, headache, weakness, muscle soreness, etc. In the second stage, on the 10th day, the person is vomiting blood or diarrhea, he is having a high fever, he is having extreme fatigue. In the third stage, that is in the 11th day, there is a brain damage, there is bleeding from nose, mouth or anus. And in the fourth stage, which is happening on the 12th day, there is the person undergoes comatose also. It could cause organ failure also, shock, massive internal bleeding, and eventually the person dies within 12 days. Now Ebola is actually becoming a dreadful disease, specifically in African countries. Now as you can see, this is a fact sheet of September 2014. The Ebola virus disease, also called Ebola hemorrhaging fever, is a severe fatal disease in human. This is given by WHO, this fact sheet is given by WHO, that is World Health Organization. The virus is transmitted to people from wild animals and spreads in the human population through human to human transmission. The average EVD case fatality rate is almost 50%. Case fatality rates have variation from 25 to 90%. The first EVD outbreak occurred in remote villages in Central Africa near tropical rainforest, but the most recent outbreak is in West Africa. Community engagement is the key to successful controlling outbreaks. Good outbreak control relies on applying a package of interventions. Early supportive care with rehydration, symptomatic treatment, improve survival. However, as you had seen, lots of cases of death have occurred in Africa. There are currently no licensed Ebola vaccine, but 2 potential candidate vaccines are still undergoing trials and evaluations. Now to date, the virus has killed almost 2,600 people, As you can see here on the right hand side, these are the health workers who are actually carrying the body of a woman who has just died of Ebola. Some epidemiologists to projected between 77,000 to 2,77,000 cases by the end of 2014. They say that cases can vary between 77,000 to almost 2,77,000. Now, the ease with which it is transmitted through contact with bodily fluids include saliva, sweat, urine, blood, semen, etc. Now, the threat of mutation is also lingering. The prominent figures have expressed serious concerns that this disease can actually go airborne, and there are many other mechanisms through which mutation might make it much more a dangerous disease, much more a transmissible disease and a real threat in the coming future. Now, as you can see, the major Ebola breakdown or outbreak came up. This is the 2014 data. The major was in Sierra Leone as well as in Sudan, as well as Democratic Republic of Congo and Uganda, and of course Gabon and Congo also. The total deaths were, numerous deaths were there, but a lot of people also got infection, and as you can see the number of deaths was also very high in these areas. Now, Ebola vaccine trials have started. This is a BBC news of 2014. The trial of an experimental vaccine against Ebola is beginning in Oxford. The first of the 60 healthy volunteers have been injected with the vaccine. It contains only a small portion of the genetic material from the virus. So, technically this vaccine itself cannot cause any disease, because we are using a little genetic material from the virus and not the virus as a whole. Normally, it would take years of human trials before a complete vaccine is approved, but in such a case of urgency, generally, specifically for example, how the outbreak took place in West Africa, experimental vaccine is, the trials are running at astonishing rate. So, if the trials are successful, it would be used to immunize health workers in the affected areas from the coming years. Now, this is the R&D map of vaccine R&D map of India, where you can see these are the places where mostly vaccine development is taking place. It also includes NII Delhi, IMT Chandigarh, AIIMS Delhi, JNU Delhi, and many other places, of course IPM Hyderabad, IIL Hyderabad, many other areas where vaccine R&D is taking place. Now, with this I would like to end my lecture, and again I would like to say that bhul na jana, teekakaran zarur karana. Thank you.
With this note, thank you ma'am, thank you so very much for wearing the masses, and yes, whatever said by Dr. Hardeep Kaur is very, very beneficial for you. So, keep watching the lectures, as the lectures are already there for you on YouTube, and this lecture is also going to be uploaded on YouTube very soon for you. For any feedbacks, questions or suggestions, you can definitely mail us at info.cc@nic.in. We would be meeting again very soon. Till then, take care. Goodbye. Thank you ma'am. Thank you so very much.